Philip again for inviting me to be part of this wonderful initiative. I really think that it's a really great thing, and I'm happy to share my data today. So today I'm going to talk to you about macrophages and how they regulate metabolism beyond the classical role in inflammation. And I'm going to talk to you about two studies mainly that we have published, and I'm also going to present some unpublished data. So I would like to start with a brief introduction on macrophages. So macrophages were discovered in the late 1800s by Ilya Mechnikov, and he discovered these cells as cells that were able to eat up big particles, and that's why they call macrophages, because macro comes from the Greek big and phage from eater. And you can see in this little movie here, for example, these macrophages eating up some bacteria. So this was the main function described for the macrophages. And since then, they've been extensively studied, and there are a lot of roles that have been described for macrophages. First, mainly in the immune regulation, in immune response, for instance, antigen presentation, antimicrobial actions, and antigen and antibody uptakes. It's just some of them. And also more recently, in tissue homeostasis. So they regulate tissue remodeling, for instance, by phagocytosing, so eating up dead cells. They can also contribute to angiogenesis, which is the generation of new vessels, or wound healing by repairing tissues, for instance. And phagocytosis, as I said, is the main role of macrophages, and that can contribute to both the immune regulation as well as tissue homeostasis. And so I said they've been discovered more than a century ago, so but there's still a lot of surprises with these cells, and that's why I particularly like them. So for instance, recently, there was a group in France that discovered that macrophages were involved in the reason why tattoos are staying in the exact same place your whole life. So that's kind of why you have to be careful when you get a tattoo, because it's going to stay there and it's not going to move. And the reason is because you have macrophages in the skin, they call dermal macrophages, that actually phagocytose the ink, and they stay there and they're renewed. There's a new macrophage coming and staying also exactly there. So I think that this is a really surprising role of macrophages, and that's just to show you that there's so much to discover about these cells. So I wanted to give a simple example of what macrophages are most well known for. As I said, they are mostly involved in the immune response, in the inflammatory response. So we're going to take a simple example. So let's say that you have an injury here and you have bacterial invasion. The first cells that respond to this injury are mast cells. And mast cells are going to release some factors called chemoattractants. So they attract other cells, and they will attract cells that are going to come and pretty much remove the bacteria by phagocytosing the bacteria. And the first cells are the neutrophils, and then after that come the macrophages. And these macrophages are what we call pro-inflammatory, or M1 macrophages. So they're going to also contribute to phagocytosing the cellular debris and the pathogens, and they produce a lot of what we call inflammatory cytokines. One of the most well-known is, for instance, TNF-alpha. So this is the reason why, because of these inflammatory cytokines and all these inflammatory cells coming to the site of injury, that you have pain, you have swelling, and you have an increase in temperature. Right after that, you have something that is called the resolution, where you have, as I said earlier, neutrophils dying, and then the macrophages that this time are called anti-inflammatory, or M2 macrophages, are removing these dead cells, and they're also producing both anti-inflammatory cytokines to resolve the inflammatory response, And they're also going to produce growth factors to promote tissue repair and wound healing. So if we look at a simplified kinetic of the inflammatory response here, we can call this type of example an acute inflammatory response, because you have the stimulus here, which was the injury and the bacterial invasion. You have an increase in the production of inflammatory cytokines, and then the resolution with the production of anti-inflammatory cytokines and growth factors by anti-inflammatory macrophages. So then you have tissue repair, and that's why it's called acute inflammatory response. However, if there is an impairment of the resolution, if there's a failed resolution, then you have what we call chronic inflammation. And that is the case in chronic inflammatory diseases such as rheumatoid arthritis, Crohn's disease, and psoriasis. So these are inflammatory diseases. And most recently, obesity, associated insulin resistance, and type 2 diabetes were also described as chronic inflammatory diseases. However, I do have to say that they're not the inflammation is not at the same level as, for instance, rheumatoid arthritis. We're talking about low-grade inflammation here, but it's still chronic inflammation. So we're going to talk a little bit about that here. And we're going to first start by talking about obesity. Obesity is a major health issue. As you know, there is a high prevalence worldwide, and it's rapidly increasing. And the reason why it has to be taken seriously is because obesity is a high risk factor for many diseases. Through this seminar series, we heard, for example, that obesity presents a risk factor for cancer, for instance. And in my lab, we are interested in insulin resistance and type 2 diabetes, as well as non-alcoholic fatty liver disease, which is the accumulation of fat in the liver that is often observed in patients that are obese and insulin resistant. The interesting thing is that not all obese patients are insulin resistant. About 25% of people are insulin sensitive, and we call them metabolically healthy. What it means is that they might have other problems, but at least they are insulin sensitive when they matched for BMI. And my lab is pretty much interested in this black box here, in trying to understand the cellular and molecular mechanism involved in the development of insulin resistance in obesity. Or in other words, why are some obese people becoming insulin resistant while some others are staying insulin sensitive? And one of these mechanisms, or one of the mechanisms that I described, is inflammation. And here are some of the reviews that are describing that. And one of the reasons why inflammation has been suggested as a link between obesity and insulin resistance is the discovery of immune cells in the adipose tissue of obese insulin-resistant patients. In the 93, Gökhan and Ostamistigi actually described that the expression of inflammatory cytokine was increased in the adipose tissue of mice and human with obesity and insulin resistance. And later on, it was in 2003 that the lab of Tony Ferrante discovered that macrophages were actually present in the adipose tissue of mice and human with insulin resistance. And here in these figures that you see here, you have two cohorts that are matched for BMI, so they are the same level of obesity. And on the left side here, you have the insulin sensitive, and on the right side, the insulin resistant. And when you look at the omental adipose tissue of these people, so the visceral adipose tissue, you can see here that you have some dense area next to the adipocyte. So these are the adipocytes, and you have this dense area. And these are actually immune cells, and particularly macrophages. So in human, and as well as in mice, you have accumulation of macrophages in the adipose tissue of that is associated with insulin resistance, actually. So then macrophages have been extensively studied and to understand their role and their contribution to insulin resistance, and particularly adipose tissue macrophages. And I'm just going to summarize the data here from the literature as well as our data. And the bottom line is that macrophages are not either good or bad, and either inhibiting or promoting insulin sensitivity. It is a question of yin and yang and balance within the tissue. And adipose tissue macrophages, if we summarize all the data, can contribute to insulin resistance, but also promote insulin sensitivity. And more recently, we have accumulated data actually showing that the same macrophage can produce factors that are both beneficial and detrimental to insulin sensitivity. One of the important roles of the adipose tissue, maybe the most important role, is to store nutrients in forms of fat. And in people or animals that are obese and insulin resistant, the storage capacity of the adipose tissue, so the ability of the adipose tissue to store excess nutrients in forms of fat, is decreased. And because it's decreased, the lipids that are not stored by the adipose tissue are being stored by non-adipose tissue, such as liver. And you have what we call fatty liver disease or non-alcoholic fatty liver disease. And this is because you can see here the accumulation of fat in the liver in people that are not drinking excessive amount of alcohol. And these lipids in the liver are considered toxic, and they could contribute to insulin resistance in the liver as well as systemically. So we decided to study the role of macrophages in the liver, and particularly their role in the development of insulin resistance in obesity. And the prevailing paradigm until last year was that macrophages in the liver in obesity are in a pro-inflammatory state. So they go from healthy anti-inflammatory macrophages into a pro-inflammatory phenotype, and these pro-inflammatory liver macrophages would produce excess amount of inflammatory cytokines that are then blocking insulin sensitivity in liver, therefore inducing insulin resistance. So that was the paradigm, and we decided to test this theory by using the power of next generation sequencing. So we collected macrophages from the liver of mice that were fed a normal diet. So these mice are lean, or a high-fat diet. So these mice are obese. And then from different cohorts of people, we had lean, obese, insulin sensitive, and obese insulin resistance that were matched for BMI. So we collected the macrophages, and then we performed RNA sequencing to study the transcriptomic profile. And one of the first things that we did was to look at the expression levels of genes that were known to be involved in the pro-inflammatory switch that I was talking about earlier. If macrophages were becoming pro-inflammatory with obesity and insulin resistance, some of the genes that are involved in this pro-inflammatory switch, should be regulated. So here we have a heat map, and you can see here the scale. So the darker, the most upregulated the genes are. So if the colors are similar, it means that there's not really a regulation of the genes that we're looking at. We compared lean and obese human, and we compared obese matched for BMI, insulin sensitive, and insulin resistance. And here we have the mice that were either lean or obese. To make a long story short, you can see here that you have similar colors. So so it means that there was not a regulation of the genes that we looked at. And what it means is that at least at the transcriptional level, we couldn't detect a pro-inflammatory switch in liver macrophages in obesity and insulin resistance. So we then did a lot of experiments. We looked at the protein level. We also manipulated genes that are important for inflammation and immune activation of macrophages. And in summary, we found that liver macrophages do not actually become pro-inflammatory in obesity. And therefore, we do not think that liver macrophages contribute to insulin resistance through the production of inflammatory cytokines. So we thought, well, if we want to understand what macrophages are actually doing, we need to really understand their phenotype. And to do that, we decided to use a system biology approach where we used multiple techniques within a platform where we have lean and obese mice, lean and obese individuals, insulin sensitive and insulin resistant. And after careful metabolic phenotyping and liver disease assessment, we collect blood and liver biopsies. We then perform, as I told you earlier, RNA sequencing. We also looked at the small RNA profile by doing a small RNA-seq, and we used another technique called global run-on sequencing to look at the active transcription of gene. Because macrophages are very plastic and they're heterogeneous, they can acquire multiple phenotypes even within the same tissue. We're also using single-cell techniques, such as single-cell RNA-seq. And I'm going to talk to you about that a little bit later. But here, we just have an example of the multiple clusters, so the multiple cell population that are present in the liver of lean and obese mice. And as I said, we're going to talk about that later. We're also looking at the proportion of macrophages, and to do that, we're using flow cytometry, typical flow cytometry, or mass cytometry. So mass cytometry is similar to flow cytometry, except that instead of having antibodies that are labeled with fluorophore, here they are labeled with heavy metals. So we can use up to 45 antibodies. And this we're doing in collaboration with Florent Ginou in Singapore. And here is also an example of a plot where you have in different colors the multiple population that are present in the liver of obese mice. We also looking at the location of these macrophages, because the microenvironment is also very important for the type of phenotype that the macrophage can acquire. So we're using techniques such as in situ sequencing, particularly padlock in situ sequencing, where we can look for up to 200 genes in one single section. We're also using the classical RNA in situ hybridization and immunofluorescence. And finally, the cellular metabolism of any cell, but in this case, we're looking at macrophages, is very important to define the phenotype. So we do that in situ because it's very important to not manipulate the cells too much when you're looking at cellular metabolism, because it changes quickly. And we're doing that with, in collaboration with Arvind Hashemi at University of Vienna. So we put this platform together pretty much to understand the phenotype and the role of macrophages in the regulation of metabolism. So we're going to go back to the RNA sequencing. I told you earlier that macrophages are not becoming pro-inflammatory with obesity and insulin resistance. So the next question is anything happening to these macrophages? And the answer is yes, because as you can see here, we had about more than 1,000 genes actually that were significantly regulated when we compare the mice that were fed a high-fat diet versus the normal diet. So this is a diet-induced model of obesity. Or when we compare OB-OB mice to wild-type mice. And the OB-OB mice are a genetic model of obesity. They're pretty much lacking the satiety hormones, so they keep eating all the time. And that's why they become obese. And we also had a few RNAs, microRNAs that were significantly regulated, and we're going to talk about that later. So if we look now into details into these genes that were regulated, we decided to look at genes that were specifically expressed by liver macrophages compared to hepatocytes. So we looked at the whole liver actually, because the whole liver is pretty much, hepatocytes. 80% of the liver is hepatocytes. So here again, we have this heat map that I was showing you earlier. The darker, the higher the expression of the gene is. So here we have genes that are specifically expressed by liver macrophages, and also that are significantly upregulated with high fat feeding, so with obesity. And the top gene was a gene called IGF-B7, or insulin-like growth factor binding protein 7. And it had been studied in the regulation of insulin signaling, but mostly in Drosophila. So we were interested in this gene. It is a secreted protein, so we looked at the release of IGF-B7 from liver macrophages isolated from mice that were fed a normal diet or high-fat diet. And as you can see, there is an increased release of IGF-B7 in liver macrophages from obese mice. We then looked at the expression of IGF-B7 in human liver macrophages and also in Drosophila, because as I told you, it was shown to be expressed in Drosophila. So here in green, we did the RNA in situ hybridization, and you can see the IGF-B7, and in red, it's a macrophage marker called EMR1 or F480. And you see that IGF-B7 is also highly expressed in human liver macrophages. Now, in the Drosophila, these red dots here are IMPO2, which is the homolog of IGF-PP7. And we did this experiment in collaboration with Adam Bashgar at the University of South Bohemia in Czech Republic. What had been shown before was that IGF-P7 was expressed by cancer cells and regulating insulin signaling. But what we have shown is that IGF-P7 was actually expressed by hemocytes, which are the immune cells of Drosophila. So the next question, after the fact that macrophages do not become pro-inflammatory with obesity and insulin resistance, there is something happening to these macrophages because we saw that genes were significantly regulated, and then we found that IGF-P7 was the gene that was the most upregulated in liver macrophages. And so the next question was, what is the role of IGF-P7 expressed by liver macrophages in the regulation of metabolism? And the only way to answer that question is actually to manipulate IGF-P7 expression only in liver macrophages, not in the rest of the cells in liver and in the cells in the rest of the body, but only in liver macrophages. So in order to do that, we used a technology called glucan-encapsulated RNAi particles, or GURPS. It is a technology that I invented during my postdoc, and it's a technology that allows the delivery of small interfering RNA, or siRNA, to macrophages to silence gene in a tissue and a cell-specific manner in vivo. So I'm going to explain a little bit what these particles are. So here you can see, this is a scanning electron microscopy actually, where you have peritoneal macrophages that are treated in vitro with the particles. These are pseudocolors, and in green you have the particles bound to the macrophages or on the way of being phagocytosed, and some of them actually have been already phagocytosed. So this technology is based on baker's yeast that are used to make bread or beer. And this is the yeast cell wall actually. So you see the different layers that are composing actually the yeast cell wall. And through chemical extraction, we're removing most of these yeast cell wall components, and we end up with these hollow porous shells here that are made with one of the component of the yeast cell wall, called beta-1,3-D-glucan. And we can actually load this particle with siRNA. And very briefly, we use a peptide called endoporter that is positively charged and can bind nucleic acids such as siRNA that are negatively charged. And so we're making these complexes through electrostatic interactions, and then we can load these complexes into the particles. And you can see here a red siRNA that is bound to the particles. They're pretty big. They're about 2 to 4 microns in diameter, so they can only be phagocytosed. So that's the first layer of specificity. Only macrophages can phagocytose. I mean, there are other cells, but the cells that are professional phagocytes are macrophages. The second layer of specificity is the fact that the glucan can be recognized by receptors, such as the dectin-1 receptor, that is expressed by macrophages. So that's the second layer of specificity. And then depending on the model that is used and the route of administration, we can target macrophages in different tissues. So here I'm going to show you the efficacy. So this is, again, the same image that I was showing you earlier. So when we use the particles in vitro and we target SPP1, the target doesn't really matter here, it's just to show you the efficacy. As you can see, when we load the particles with an siRNA targeting SPP1, we had almost a knockout compared to particles that were loaded with a scramble, non-targeting siRNA. So that's the control. When we did an IV injection and we isolated liver macrophages of obese mice, and here you can see FITC labeled particles within macrophages that are stained in red, and you can see that we had a significant knockdown of our gene of interest, IGF-7, compared to the control. And finally, here we have another example. This is after an IP injection in an obese mouse, and these are adipose tissue macrophages that are stained in red again, and you can see the FITC particles inside the macrophages. And we had a strong knockdown of our gene of interest here, SPP1, when the mice were injected with GURPS loaded with a targeting siRNA compared to the control. So if we go back to our question, what is the role of IGF-7 expressed by liver macrophages? We loaded particles with siRNA targeting IGF-7. We fed the mice for 8 weeks with high-fat diet, and then we treated them for 2 weeks. So first, I would like to show you that when we use particles that are here labeled with a radioactive probe, iodine-125, and we do live imaging, as you can see, the particles are found in the liver, and that's true for up to 7 days. I mean, we looked up to 7 days. When we used FITC labeled particles and we isolated from the same liver, the liver macrophage fraction, here stained with the F480, which is a macrophage marker, and the hepatocytes stained with albumin, which is an hepatocyte marker, you can see that all the particles here in green are seen within liver macrophages. So we compared mice that were treated either with particles loaded with an siRNA targeting with IGF-7, targeting IGF-7, sorry, compared to control. And as you can see here, we had a significant knockdown of IGF-7 in the liver macrophage fraction. And we had actually no difference in the expression of IGF-7 in the hepatocytes. And that is because, well, the hepatocytes do not contain particles, so that's why you don't have any knockdown. So we really have specificity in the liver macrophages. And we have published before that the particles using this method are really targeting just liver macrophages. Then we looked at the metabolic status of the mice, and I'm showing you just here a few parameters. First, we looked at fasting glycemia. And as you can see here, when we knocked down IGF-7 only in the liver macrophages, the mice were becoming normoglycemic. So they were going from being hyperglycemic to normoglycemic. The most dramatic effect, actually, of knocking down IGF-7 in liver macrophages was to decrease the content of liver in triglyceride, in lipids. So we had about a 50% decrease in liver triglyceride. And as I told you earlier, the accumulation of lipids in the liver is really important. So that's why it was really important to look at that. And here we have electron microscopy images. And as you can see, when we knocked down IGF-7 in liver macrophages, we had a decrease in the size of the lipid droplets, as you can see here in the control. So I'm just going to summarize the data that we published in Nature Metabolism last year. So we did the experiment in Drosophila and in mice. I showed you mostly the mice, but what we saw is that the expression and the release of IGF-7 is increased from liver macrophages, is increased with obesity and insulin resistance. We also found that IGF-7 could actually bind the insulin receptor. and regulate and enhance the ability of insulin to activate AKT, so downstream protein AKT. And AKT increases lipogenesis and blocks gluconeogenesis in an insulin-sensitive mouse. But then in insulin resistance, we found that the ability of IGF-P7 to bind insulin receptor was decreasing. However, the binding of IGF-P7 in the insulin resistant state was enough to still activate ERK and phosphorylate ERK. And therefore, we had the downstream concomitant and pathological activation, induction of lipogenesis and gluconeogenesis, leading to hepatic steatosis, which is the accumulation of fat in the liver, and hyperglycemia. We then looked at the expression of IGF-PP7 in liver macrophages in human, and as I told you earlier, we actually saw that IGF-P7 was expressed by liver macrophages, but we were disappointed to see that IGF-P7 was not regulated with obesity and insulin resistance in liver macrophages. However, we discovered something interesting. In collaboration with Claudia Kerr, we looked at the conservation of IGF-P7 gene, and as you can see, it was pretty conserved. But one thing that we saw that was different among the species is the RNA editing of IGF-P7. What we found is that IGF-P7 could undergo RNA editing called A2I RNA editing. So I'm going to explain a little bit what A2I RNA editing is. So you have these enzymes that called the ADARs, and they are deaminases. So they can remove an amine group. And here, that's what they do. From an adenosine here, they remove this amine group to change it into an inosine. And this inosine is read by the translational machinery as a guanosine. RNA editing happens a lot, and most of the time in non-coding region, and it doesn't always lead to a change in the amino acid sequence. But in this particular case for IGF-P7, the RNA editing was happening in the binding region, and as you probably remember, I told you that IGF-P7 could bind insulin receptors, so the binding domain is really important for its function, and it was leading to a change in the amino acid sequence. When we looked at the proportion of the frequency of the RNA editing, as you can see here in this table, it was happening in 30% of the lean patient, 30% of the obese insulin sensitive patient, but in 90% of the obese insulin resistant. We then looked at the frequency, the allele frequency of the RNA editing. So in other words, how many transcripts get edited when it does get edited. And in the lean and obese insulin sensitive, even in the patients that had RNA editing, it was at a low frequency. But in the obese insulin resistance, it was not only happening in most of them, but also at a high allele frequency. So now the question was, what is the effect of editing on the function of IGF-P7? As I told you earlier, IGF-P7, at least in mouse, we could bind the insulin receptor. And what we found is that in an obese person that is insulin resistant, the IGF-PP7 could bind the insulin receptor. As we saw in mice, the binding capacity was reduced in insulin resistance, but still enough to activate ERK, not AKT, but ERK. And then we had the concomitant induction of lipogenesis and gluconeogenesis, leading to steatosis and hyperglycemia. However, when IGF-P7 was getting edited, it was increasing its ability to bind the insulin receptor and therefore enhancing the ability of insulin to phosphorylate AKT. And therefore, we speculated that the RNA editing of IGF-P7, which was only happening in human, not in mice, could be a protective evolutionary mechanism to counteract insulin resistance. And we are really interested now in the whole RNA editome, and that's something that we're looking into. I'm going to show you some preliminary and unpublished data. So the first thing that we looked at was, we wanted to look at another species closer to humans. So we looked at macaques. And we compared all types of RNA editing. I told you a lot about A to I, but there are a lot of different types of RNA editing. And we compared these different species, the frequency of RNA editing in liver macrophages. And as you can see, the A to I, the one that is happening for IGF-7, is the most frequent in human. But RNA editing in general was more frequent in humans compared to macaques and mice. We then compared liver macrophages to hepatocytes in human because we wanted to know if it was something that was more specific to liver macrophages. And indeed, we saw that the RNA, all types of RNA editing, and particularly A2I, was higher in liver macrophages compared to hepatocytes in human. And then we compared the obesity, insulin sensitive, and insulin resistance. We saw earlier that IGF-7 was more edited in the insulin resistant, but then here we're looking at the whole RNA editome. And indeed, we saw that RNA editing, something that was occurring more in the liver macrophages from obese insulin resistance compared to obese insulin sensitive patients. So now our question is, how is the RNA editing getting regulated? Why it is happening in a cell-specific manner? Is it happening in particular macrophage population? And more importantly, what is the relevance of RNA editing to metabolic diseases? All right, so I talked to you about the fact that macrophages were not becoming pro-inflammatory with obesity and insulin resistance in the liver, in human, in Drosophila, and in mice. I showed you that they can produce non-inflammatory factors such as IGF-7 that can directly regulate metabolism. But the question was, we had all these genes, of thousands of genes that were regulated. So what are these other genes? What are these, what is the phenotype of macrophages with obesity and insulin resistance? When you have RNA sequencing data, you pretty much have two choices. Either you go with the gene that is the most regulated, that's what we did with IGF-7. The other way to analyze the data is to look at pathways that are regulated. So even if genes are not highly regulated, but a small regulation in a lot of genes that belong to the same pathway could have a dramatic impact. So when we looked at that, we looked at the rest of the genes. I'm just summarizing the data here. We saw that genes involved in oxidative stress were highly regulated in liver macrophages with obesity and insulin resistance. That was true for mice and for human. And we were not that surprised because, as I told you earlier, the storage capacity of the adipose tissue with obesity and insulin resistance is reduced. And that leads to the ectopic deposition of lipids in non-adipose tissue, such as liver. And this leads to fatty liver disease. And in order to handle all these lipids, you have an increase of lipid peroxidation to burn out these lipids. And that leads to an excessive production of reactive oxygen species, or ROS. And that's what leads to oxidative stress. So here we looked at oxidative stress. And you can see here that we measured reactive oxygen species in the medium of liver macrophages. And you can see that it was increased with high fat feeding, so with obesity. We also looked at the whole mouse liver, and we had an increase in a component called malonyl-dihaldehyde, which is a byproduct of lipid peroxidation. And you can see that it was also increased. And then we looked at the human livers. We looked at both reactive oxygen species and reactive nitrogen species. And you can see that we had an increase with obesity and particularly with insulin resistance. So in mice and in human, we had the increase of oxidative stress in the liver with obesity and insulin resistance. So usually in a healthy cell, what happens is that when you have oxidative stress, there is the endogenous antioxidant response to pretty much scavenge the excess reactive oxygen species. And this is regulated by a transcription factor called NRF2. So in physiological condition, when you don't have oxidative stress, NRF2 is bound to another protein called KEAP1. And KEAP1, the binding of NRF2 to KEAP1 induces its degradation. And when you have excess levels of ROS in the oxidative stress condition, KEAP1 is not bound to NRF2, and therefore NRF2 is not degraded and can translocate to the nucleus and induce the expression of genes that are able to encode protein that are going to scavenge reactive oxygen species. So that's why it's important for the antioxidant response. So we thought, since oxidative stress is increased, we're going to look at the antioxidant response. And we were pretty surprised, because when we looked at liver macrophages from high-fat diet mice, we looked at hepatocytes from high-fat diet mice, and mouse liver in 2 models, as well as human. We saw that there was a dramatic decrease of the protein levels of NRF2 with obesity and insulin resistance, as you can see here in this Western blot. So it was completely gone with insulin resistance. So we thought that, well, the antioxidant response could be impaired with obesity and insulin resistance. And we thought, if NRF2 is decreased, maybe it's through KEAP1, binding to KEAP1. So we looked at that. And actually, what we saw, and I'm not going to show you the data, I'm just going to summarize them, is that in obesity and insulin resistance, NRF2 was not regulated either at the transcriptional level, so the mRNA level was the same, and it was not regulated through KEAP1. So we thought there must be another post-transcriptional mechanism regulating the protein level of NRF2. So we decided to look at microRNAs, because microRNA can also regulate the protein level of any protein that you're looking at. So we did small RNA-seq. We used liver macrophages from lean and obese mice. Here I'm just showing you OB, but we also did a high-fat diet. And we saw that there was a significant increase of a microRNA called Myriam-144. And Myriam-144 actually can target NRF2. So one of the speculated targets was NRF2. So we looked at its expression in the whole liver in mice that were fed a high-fat diet, and you can see here that it was significantly increased. It was also increased in the OB, in the other model of obesity. We then looked at, to confirm the RNA-seq data by qPCR, we looked at the liver macrophages, and you can see here that it was significantly increased. And we also looked at the human livers, and you can see that the expression of Myriam-144 was increased with obesity, and particularly with insulin resistance. So we decided to look into Myriam-144. So we knocked down Myriam-144 in liver macrophages using the particles, because, as I told you, we can pretty much load any nucleic acid. And here we loaded antagomeres, which are inhibitors of microRNAs. And when we knocked down Myriam-144 here, we had, I mean, it was almost a knockout. We had an increase in the protein level of NRF2, as you can see here, in liver and liver macrophages. And we had a decrease in the levels of ROS in the whole liver following knockdown of Myriam-144. And we also had a decrease the release of reactive oxygen species from liver macrophages and from hepatocytes following knockdown. We also want, since Myriam-144 was also increased in patients with obesity and insulin resistance, we wanted to use an in vitro model. So in collaboration with Volker Lauschke, we cultured the liver or spheroids or liver organoids. So they were composed of human non-parenchymal cells. So these are everything macrophages, endothelial cells, all the immune cells, in addition to human hepatocytes, and we can culture them in 3D, as you can see here. And by using a media that contains high levels of lipids, free fatty acid, insulin, and glucose, we can mimic fatty liver disease. As you can see here, we had accumulation of fat. You can see the lipids in green here within the spheroids. And we could also induce insulin resistance. So here are liver steroids that are treated short-term with low concentration of insulin, and you can see that we had increase in the phosphorylation of AKT. But then when we treated the steroids with high concentration of insulin and long-term, as you can see, the phosphorylation of AKT is completely blunted. So we do induce insulin resistance. So we could mimic in vitro steatosis and insulin resistance. So when we looked at oxidative stress, so we treated, as I said, the steroids with high levels of free fatty acid. As we saw in the whole liver, we had an increase in the expression of Myr144. And we had an increase in the levels of reactive oxygen species, as you can see here. And when we knock down Myr144 here in the non-parenchymal cells, we blocked actually the ability of free fatty acid to increase reactive oxygen species. So we reduced oxidative stress. So we confirm pretty much what we saw in mice in the liver organoids. So in summary, what we saw is that, and we did more experiment to look at actually the regulation of Myr144. And what we found is that when there's excessive amount of reactive oxygen species, that leads to the activation of ERK, the phosphorylation of ERK. And ERK is going to phosphorylate and activate a transcription factor called GATA4. And GATA4 can induce the expression of Myr144, and Myr144 then reduces the level of NRF2. And that leads to even more ROS being released and inducing then the phosphorylation of ERK in hepatocytes, the activation of GATA4, and this vicious cycle where you have oxidative stress in the cells. And that was in the liver, and that was true for mice and for humans. So far, the literature has, most of the literature has described macrophages as bulk, and including the studies that I presented. So you either have the good macrophages and the bad macrophages, the good macrophages that stimulate insulin sensitivity, and the bad macrophages that contribute to insulin resistance. But it's a little bit of a simplistic view considering the plasticity and the heterogeneity of macrophages. So just to give you an analogy, it would be like if I was giving this talk in a seminar room and I had to describe the people in the room, and I would decide to just do two groups with males and females, let's say 50% males, 50% female. Well, that would be a little bit of a simplistic view because obviously people are much more complicated. So now if I had to make many groups and compare this group according to the characteristic, the many characteristics, then it would become much more difficult to do that. And that's kind of the same thing if you're taking a tissue and all the cells that are, all the macrophages that are present in the tissue. If we just say, well, these macrophages are good because they contribute to insulin sensitivity and these are bad, that would be a little simplistic. But if we start describing them according to the phenotype and we're looking at the transcriptomic profile, then there are tens of thousands of genes that could be regulated. And we have the ability now with single cell sequencing and computational tools to pretty much condense all these different dimensions into 2 dimensions. And this is what we did. We performed single cell RNA sequencing of non-parenchymal cells. So as I said earlier, they have the macrophages, but also all the rest of the cells except the hepatocytes, from lean and from obese patients that are insulin sensitive and insulin resistant. And we also had the mice. We stain these non-parenchymal cells with multiple antibodies, and then we sort them, but in an unbiased way, meaning that we don't sort them according to the staining. We just sort them one by well in this multiple well plates, depending on what you're using. But then we can go back and we can look at the staining in each well, and it's called index sorting. And then we perform single-cell RNA-seq using the SMART-seq2 protocol, or a modified version of that that is adapted for non-parenchymal cells. So as I told you earlier, we're going to go into that a little bit into more details. Here, each cluster represents a cell population, and they pretty much cluster together because the transcriptomic profile is very close. So here we had all the different populations that are present, or the one that we were able to sort that were present in the liver of lean and obese mice. And we have the ability to extract data from only the macrophages and recluster the macrophages. And that's what we see here. We had 3 populations of liver macrophages. And that was true for the lean mice and the obese mice. And now if we look a little bit more into what these populations are, first we looked at markers of resident liver macrophages, or Kupffer cells. One of them is CLEC4F. And as you can see, it was highly expressed by this population here, the LM3. So we believe that these are the resident liver macrophages of the Kupffer cells. And we had another population that was actually expressing IGFBP7. And that was really interesting because the data that I described to you earlier, where we knocked down IGFBP7 and we saw an effect on the phenotype of cells or spheroids, is probably not due to the decreased expression of IGFBP7 in all the macrophages, but only in one population. So this population could be regulating steatosis and insulin resistance by itself. And the third population is a really interesting population because when we looked at the number of genes that are regulated upon obesity and insulin resistance, we saw that this population is the most affected by obesity and insulin resistance. And what we saw actually is that one of the pathways that were regulated is oxidative stress. So maybe again, the data that we collected on oxidative stress in liver macrophages could be due to only one population, and maybe this population is regulating oxidative stress in the whole liver, and that's what we're looking into right now. One of the genes that was the top gene expressed by this population was PLAC8. It's not that is that important, but it's just to find a way to detect this population when we do in situ, and that's what we did here. We stayed with the RNA in situ hybridization PLAC8, and we saw that this population was found around the vessels, actually, or within the vessels, and it was not particular vessels, it was all the vessels in the liver. So right now we studying the role of this population in the regulation of liver function. So this is the take-home message. It's pretty much that macrophages play multiple roles, or even opposite roles within the same tissue, metabolic tissue, and they can regulate homeostasis and metabolism with these different roles. This is my team. Here we have Valerio Azimato. He's a postdoc, and he is the one who led the project on oxidative stress and liver macrophages. Cecilia Morgantini was and is still the lead author for the Nature Metabolism, where she described the IGFBP7. Now she's working on the RNA editing. Emily Barabi is a PhD student, and she's working on the heterogeneity of liver macrophages in obesity and insulin resistance. And these are my new people, my new postdoc, Sebastian Nock, who is going to now work on the role of liver macrophages in NASH. And Ping Shen is involved, as you can see here in her picture, in the computational analysis of all these different projects. And she also has her own project that she's starting now. This is my lab manager, Laura Levy, and the lab technician, Myriam Aouadi, that just joined the lab also. This is my former lab members, actually, that contributed to the study that I presented. I would like to also thank my wonderful collaborators that contributed to all the work that I presented and some of the work that is ongoing currently in my lab. And these are the funding agencies that, of course, also contributed to this study. And I would like to now thank you for your attention, and thank you for joining in. And I'm done. I just stopped sharing. I'm not sure what I'm supposed to do.